So good morning, everyone. I, Dr. Poonam Joshi, welcome you all once again in our Saturday academic program. Today lecture will be taken by Dr. Manjunath. Dr. Manjunath is a professor in Department of Pharmacology, Tata Memorial Center, ACTRAC. So I would request Dr. Manjunath to go ahead with the presentation.
Good morning, everybody. Thank you, Dr. Poonam. So, shall I start?
Yes, please start, Dr. Manjunath.
So, what I was told to is to give a small lecture or presentation on the phases of the clinical trials. So, rather than that, I'll just share with you what are the different clinical trials that in an oncology can be undertaken, whether it will be an observational or an interventional. So, when we tell it is like an clinical trial. So what is it? It's a type of research wherein we try to find an answer for a scientific question. Okay. So it may be like with respect to medication, device, or even any like training methods also. So there will be like different types of clinical trials, which will be like a natural history, or the prevention trials, the screening trials, or the diagnostic trials, or the quality of life, and finally an interventional trials. So natural history means what will happen if it is left untreated, or if it is treated with a standard therapy. These also can be like, these are also like a one type of the trials. The other trials are like a chemo prevention trials. These can be like an action studies or the agent studies. Action studies means do something and to prevent the cancer. And the agent studies are like take something, you give some medications, that is a prevention, chemo prevention, like the tamoxifen or the finasteride, even the aspirin for prevention of different cancers. The others are like the screening trials, for which we will be using like different methods or the techniques to use the tissue samples or the laboratory tests, imaging tests, or even the physical examinations. Next is like, I'll just give an example of this screening test or the early detection trials. This trial has been started way back in like what we call the PLCO trial, which is a screening trial, prostate, lung, colorectal, and ovarian cancer. So wherein they have started way back, and they enrolled the patients and they have followed the patients. Even in the 2015 also, like they are still continuing to evaluate on what's happening to these patients. They have collected the biologic materials and are regularly following the patients. Okay. No, so what they did is like in different conditions, like as I said, like it is in the prostate, lung, okay, and ovarian and colorectal cancers. So they have taken the baseline information from them. They have taken some like the blood samples from them, or even the history, the basic information from the patients. Okay, so from which they are they're evaluating like what's happening over a period of time. The other trials are the diagnostic trials. So these trials usually used to develop the better tests or the procedures to identify a suspected cancer earlier or more accurately. So as you know that these diagnostic tests can be like the imaging as well as the laboratory tests. So depending on which we can design the study, it can be like an radiology based or the lab based. Next are the supportive or the clinical quality of clinical life clinical trials. And you are aware like we evaluate the patient's quality of life, for which we have like different tools, what we call them as patient reported outcomes. These are a set of questionnaires which capture the patient's quality of life in different domains, maybe like the physical activity, the social activity, okay, everything, the mobility of a patient, everything will be captured from these patient reported outcomes, or what we call them as a quality of life trials, right? The imaging trials, as I said, this can be like a diagnostic one, okay, which will be used to screen or diagnose, like even the, whether the target binds to that, whether the drug binds to the target or not. So this can be the different imaging trials. What are these are, as I said, it can be like a screening, diagnostic, or even the guiding of the cancer treatments, right? So coming to the final, that the treatment of the treatment, what are the different interventional trials? That is the treatment trials. These normally the clinical trials are divided into the 4 phases: phase 1, 2, 3, and 4. In the phase 1, they will be what they will do is they evaluate the safety and tolerability of the drug. That is that how much, what are the different adverse effects that can be seen with this drug, and the tolerability. What is the maximum dose a patient can tolerate this drug? Normally, the phase 1 clinical trials will be conducted in the healthy volunteers, except in the conditions which are used like a toxic drugs or the cytotoxics, that is the cancers, and also in the HIV patients. The phase 2 trials, they determine whether this drug is active in a specific cancer type. So this compared to the phase 1 trial, the phase 2 will be conducted in a larger number of population with a specific cancer type. And in the phase 3, they will determine whether the drug is effective or not. In a period of time, like this phase, the phase 3 will be more similar to that of a normal day-to-day practice, but it will be like a somewhat more controlled than the daily practice. The phase 3 will be conducted in a large set of patients, okay, and with a specific group of cancers, maybe also with some definite comorbidities, maybe like the mild type of renal failure, mild to moderate type of renal failure. So in a specific group of patients, they'll be carrying out these trials. After the phase 3 trial, the drug will be approved and it goes into the phase 4. So after this, once the drug is approved, what we call it as a post-trial, phase 4 trial, post-marketing surveillance. In this phase, the pharma companies will provide the information about this drug, about the toxicity of this drug to the DCGI. Okay, so as I said, the primary purpose of the phase 1 is to evaluate the toxicity and to study the kinetics. So basically, like how the drug is absorbed, distributed, or the metabolized, and it is eliminated from the body. It may provide, I repeat, it may provide some evidence of response, but it will not be a primary aim of this phase 1. They will, like, the main purpose is to define the dose limiting toxicity or the maximum tolerated dose. So we'll define the dose limiting toxicity normally. So what do you mean by dose limiting toxicity? So the toxicity which limits the further dose escalation or the dose administration to a patient. And the other endpoint or the objective is to define the maximum tolerated dose, the dose which a patient can tolerate to the maximum. Means beyond this dose, that is the maximum tolerated dose, the patients will definitely face the adverse effects. Okay. So the toxicity has traditionally been the primary endpoint for the phase 1 trials. With the emergence, however, this with the emergence of these novel, like the targeted therapies or the monoclonal antibodies, the binding of the target, what we call them otherwise as a biological activity of the molecule, that is BA of the molecule, is the endpoint for this in the phase 1 clinical trials, and with which we'll get the plasma drug concentrations as well as the target inhibition in the tumor or the surrogate tissues, right? So traditionally for the cytotoxics, the primary endpoint is the toxicity, whereas for the novel molecular targeted therapies, the biological activity is the primary endpoint for these things, for the molecules. So here the just go to the next slide. So what are the additional goals or what we do in the phase 1 clinical trials? They evaluate the newer treatment schedules and also the new drug combination strategies, as well as any multimodality treatment regimens are also tested as a part of the phase 1 clinical trials. So this will be done in a small number of patients. As I said, it is usually done in the patients for the cancers as well as the HIV drugs. And the group of patients which you take them are those who have refractory to the standard therapy. Means the patients who have been treated previously with the multiple lines of therapy and have progressed to the further, and they don't have any, as of now, the standard therapy. But we should include the group of patients who have the adequate organ functions and adequate good performance status. Okay, so normally what we do in a phase 1 clinical trial designs are like this will be like, these are the open label studies and these are non-randomized, and they are dose escalation. As I said, in the phase 1 trial, we will be seeing the dose which is maximum tolerated for the patient. Normally, we will be using the 3+3 design to find out which is the best dose that is safe as well as tolerable for the patients. Okay. So how we escalate is like what we modified Fibonacci dose escalations. Okay. So I'll just skip this. It may be like too heavy for you. Okay. Right. So what is this traditional 3+3 design? What I told you is like in this normally they will be taking 3 patients in this in a clinical cohort. In the 3 patients, they will be seeing the whether the drug is toxic or not. So we will be, as I told, the toxicity means the dose limiting toxicity. So normally the dose limiting toxicity will be defining before we start a clinical trial. These dose limiting toxicities in an oncology will be normally like occurrence of the grade 3 and grade 4 like lab parameter abnormalities or the vomitings which have been not being controlled, okay, or the diarrhea which is not getting controlled even after the standard treatment. Okay, so the dose in the first cohort, if there is no tox, this DLT is not observed in any of the 3 patients. Okay, you just see in the slide. If there is no toxicity observed in the first 3 patients, okay, they will go to the next dose level. Okay, if at least one toxicity response is seen, they will add 3 more patients at the same stage. Okay, so as I said, it will be a 3+3 design wherein initially 3 patients will be taken up. If there is toxicity in one patient, 3 more patients will be enrolled in this stage. Okay, if there are 2 or more responses, then the dose limiting toxicities or the maximum tolerated dose has been reached, and then the dose which of the previous level will be considered as the maximum tolerable dose. Okay, so this is the basic idea, that is 3+3 design, wherein 3 patients in each cohort in the first cohort, and then they will escalate accordingly. If there is no toxicities in the first group, if there is toxicities in one patient, that is a dose limiting toxicity, they go to the next, they will add 3 more patients and see, evaluate the response. So why is it like only 3 patients, or why is it like only one toxicity? Normally in the clinical trials, the toxicity should not be more than 33%. So 1 in 3 is like a 33%. So that is the reason why they'll take like a 3+3 design usually. Okay. So normally in the phase 1 trials, the normal endpoints usually kept are, as I said, the dose limiting toxicity, which is, I said, like the non-hematological toxicity or the hematological toxicity. These are usually defined as the dose limiting toxicity. And the maximum tolerated dose, that highest dose level at which less than 1/6 of the patients develop a DLT. Okay. So if, as I said, in these recent situations, the more of the like targeted therapies have been coming up rather than the cytotoxic drugs, we will be evaluating the biological activity. So how much the molecule or the drug is binding to the target will be the primary concern or the endpoint for this study, for these non-toxicity studies, wherein we'll be using, like as I said, it will be again 3+3 design, wherein we'll be using the 3 patients in this study, and then we will go to the next level if the biological activity is less. That is, the number of patients with the biological activity are 0 or 1, we'll go back to the next higher dose. If 2 or 3 patients have shown the biological activity, we'll take 3 more subjects. this goes up, means the dose will be escalated up sequentially until the maximum biological activity has been observed in 5 or 6 patients. Okay. So now the next phase is the phase 2 trials. Normally, as we said, at the end of phase 1, we'll be defining a dose which is like an suitable to go for the next level. This dose will be evaluated for the activity or its efficacy in the patients. Okay. In the phase 1 trial, we have selected the group of patients, multiple cancer types. It will be like even the breast cancer or the lung cancer, head and neck cancer, ovarian, any type of cancers who have been multiply treated will be enrolled in the phase 1 trial. In this phase 1 trial, we will be getting some idea which group of patients this drug might be useful. We will be enriching in the phase 2 those specific group of cancers. So we'll be evaluating the dose which has been like a maximum tolerated dose in a specific group of cancers, right? So here the number of patients will be more, and there will be like more of homogeneous population. Okay. And they should have like a measurable disease and will limit the number of prior treatments, right? So this normally the phase 2 trials, what we call them as a 2-stage designs. Okay, Simon's 2-stage is the like commonly used word for these trials, wherein we don't want to expose the drug to large number of wherein we don't want to expose this drug to a larger number of patients because the efficacy has not been evaluated. So a specific small number of patients will be administering them a small number of the sample size, and then we will be seeing whether there is any response or not. If there is no response in a small group of patients, then we consider the drug is inactive and we will not proceed further. On the other hand, if there is some response in a group of patients, then we'll recruit some more patients to complete the sample size, and then we'll be evaluating that whether the drug is active or not. For example, in this slide, if we see out of like 24 patients, out of 33 patients, 24 plus 9, we have evaluated a specific number like 9 patients. In this 9, there is no activity seen or the efficacy seen, then we deem the drug is inactive and we'll proceed to the, we will not proceed further. If there is more than one, I'm saying like more than one, not one, it's more than one in 9 patients, we will recruit more 24 patients and complete the study. If there are more than 3, I repeat, like in this group, more than 3 are their responses, then we consider that the drug is active and this will go to the phase 3 clinical trial. Okay. So this is like another method or another design of the phase 2 studies, wherein like what we till now we have seen like an efficacy ke baare mein. So whether the drug is effective or not. Here in this bright and day design, we'll be evaluating the safety as well as the toxicity also. Means the number of patients who respond should be more. Okay, in the phase 1, in the stage 1. Okay, in the stage 1, in a specific group of patients, the number of patients who respond should be more and the toxicity should be less. Then only it goes to the stage 2 design. If this goes to the stage 2 and has cleared, means that the drug is shown to be like more effective as well as less toxic, this will be taken up for the phase 3 trials. Okay. The other type of studies in the phase 2 are the patient enrichment studies. So you are aware like the targets, like for example, the HER2 receptors. So initially the trastuzumab was tested in a entire breast cancer patients, wherein the efficacy was not so much. So when they have done the subgroup analysis and they found that the patients or the tumors which are expressing the HER2 receptors had more efficacy, then they have evaluated in a specific group of patients or the breast cancer patients which had the HER2. and the response was far better when compared to the earlier study. So they have enriched the patients. They have selected a group of patients whose tumors are expressing the specific tumors, specific receptors. That is, we are enriching a group of population from a group of population for the trial. What we call them as an enrichment designs. This also comes under the phase 2 studies. Okay, so what are the other designs which can be considered in this phase 2 are like a low dose versus the higher dose. Okay, whether comparing the low dose in a specific in a good number of patients with the higher dose, or the placebo versus the active agent. In this second scenario, at the background or the of the existing treatment, the background of existing treatment, the newer agent will be added to a group of patients, and to the other group of patients, the placebo will be present. Okay, wherein then they will be evaluating them for the efficacy or toxicity. The other one is the randomized discontinuation design. This is also a type of phase 2, wherein the drug, that is our trial drug, will be administered over a specific period of time, and then once the stable disease, matlab the patient is responding to the treatment, and then these patients will be randomized. Means in a run-in period, we are administering the drug over a period of time and seeing the response, okay, that the disease is stable, and then we will randomize the patient into two categories, wherein the first group of patients will receive the active drug and the second group of patients will receive the placebo. Okay, so then the final analysis will be between these two groups. Okay, so that's why, and why we call them as discontinuation, because if there is no stable disease in the first stage, the patients will be dropped off. Okay, patients with progressive disease will be dropped off in the first stage itself. Over here, if the patients in this group, they are not responding, they will be switched back to the study drug. Okay, right. So when compared to the normally the phase 3 clinical trials, the stringency will be like more. Okay, means the number of follow-ups of the patient who is in a phase 2 clinical trial will be more when compared to the phase 3 clinical trial. But for the sample size, the type 1 error will be somewhat higher when compared to the phase 3 clinical trials. What we call, like normally in a phase 3 clinical trials, phase 3 ke liye, we'll take a higher power, like 90% power, and an alpha, that is the type 1 error, should be around like 0.05, that is a 5%. But in a phase 2 clinical trials, the type 1 error can go up to like 0.1, that is a 10% rather than a 5%. Okay, and the power can be like 80% or the 70% depending upon the situation. Okay, and the endpoints for this are like normally, as you said, the response to the drug, that is to be like a complete response or a partial response, stable disease or the progressive disease. Complete response means like the drug has been, the tumor has been responding, so the drug and the tumor has regressed. Partial response will be like the percentage of response, that is if the tumor is, the cancer is responding, there will be a at least like 30% improvement in the response. Okay, the disease has not progressed, it's a stable disease. Okay, the progressive means the condition has progressed to the next stage. Okay, so in the phase 2 clinical trials, mainly the limitations are like as I said, this will be conducted in a small group of patients. So if unfortunately, if there is no activity seen in a specific group, we will be considering that the drug to be like not valid, means the drug is not having an effectivity and we will drop the drug. So it's maybe a limitation for this. And the others are like the measurable disease is required. Sometimes like head and neck cancers may like specifically the demarcation of the tumor size has to be made. Sometimes it may be like not clear cut. We may not demarcate the situation. Okay. Next is a phase 3. It will be like in total. Finally, this is the main study that any company will be going to market the drug. So we will be comparing the efficacy of this drug compared against the existing treatments. Okay, and this will be like a next step of the phase 2 clinical trials. Okay. Next, the normally it will be like hundreds of thousands of the patients. It can be of a single cancer type. Okay, there should be like a well-defined eligibility criteria. There should be an internal control group wherein we will be control group means in one group we will be giving the study drug and in the other group we'll be like in giving the standard treatment. Okay, and this will be like normally as the sample size runs into hundreds and thousands, the study will be like a multi-centric study. Okay. And normally it's a randomized clinical trials and it will be like a uniform distribution, means like equal 1:1 randomization, what we call them. Okay. Next is these different trials can be like in superiority trials or the equivalence trials or the non-inferiority different clinical trials. The superiority trials means we claim that the drug is superior to the existing treatment and we'll be calculating accordingly to the sample size. Equivalence means this drug is equivalent to the efficacy of the existing drug, okay, existing standard of care, okay, with a specific margin. Okay, so we call them as equivalence trials. And the non-inferiority trials are the one the existing the newer treatment or the regimen is not inferior to the existing treatment with respect to some efficacy point, okay, but the drug is considered to be more safe, so we'll be going for the non-inferiority clinical trials. Okay, so the what we take them as a other considerations are the sample size should be prior defined, and the analysis what we use is the intention to treat analysis, means whether the what we do in the ITT is the strategy for the analysis of the randomized control trials that compares patients in the groups to which they were originally randomly assigned, whether they complied with the treatment they were given or not. Means we'll be analyzing the data according to the groups in which they were actually assigned to. Okay, and here the data that is generated from which here like the data which will be generated is very important in the clinical trial. The documentation of the data is very important. This data will be regularly checked by the data safety monitoring board or committee, right? So the different types of study designs of the normally in the phase 3 clinical trials are the parallel group designs, wherein the subjects will be divided into two groups. Here we can see like one group here, one group here. One group receives the standard treatment, other group receives the investigational treatment. In the crossover design, what happens is the drug will be given to a specific group of patients, and after a period of time, the same group of patients will receive the other standard treatments. Okay, that is the subjects will be randomized to a sequence of 2 or more treatments, wherein the subjects act or the patients will act as their own controls and make but the other thing is they can be crossover to the other treatments for the progressive diseases. Okay, this is like there's different types of designs, that is either it can be like an parallel group design or a crossover design, right? Normally the different endpoints that will be chosen are the overall survival or the disease-free survival, PFS, quality of life and symptom control, right? So just a few slides back, I told you about the quality of life, which are measured using the patient reported outcomes. And normally what we'll be using are like the EORTC QLQC 30, which is a generic version and can be used like multiple situations will be, and this captures the patient quality of life. Okay, the quality of life with respect to different domains, either the physical, psychological, okay, emotional, okay, social, all the domains will be captured using the quality of life by the questionnaires. Okay, these things, the PROs, that is the patient reported outcomes, can also be used in the situations wherein there is like a comparison of two different standard therapies, okay, and having the survival outcomes, but their quality of life may be different. For example, in a surgery, like there are two different techniques, I mean, available, but and the quality of life which may be different for these two different techniques. So these questionnaires can be used to compare these two different techniques. Okay, the quality of life can also be used, or the patient reported outcomes can also be used for the identification of negative effects of the therapy when the survival is very long, and also to find out whether a new therapy is preferable to the existing therapy, and to determine a therapeutic regimen is better than the supportive care only. Okay, so like these are the different uses of the quality of life or the patient reported outcomes. So this quality of life can be or will be a part of the phase 3 clinical trials also. And the limitations of this phase 3 are like, as I said, this will be done in a large cohort or the hundreds to thousands of the patients, and it can be like difficult to recruit, and the complex means it will be like the protocol will be very much complex, and it is like as usual the expensive. Okay, next, normally the endpoints what we choose in these oncology trials, as I said, like overall survival, that is time from randomization until the death, and they will be analyzed using the ITD, that is the intention to treat population, wherein they will be analyzed according to the group they were randomized. Others are like human assessments, that is like disease-free survival, objective response rates, all will be considered. I mean, can be considered for the endpoints for the different conditions. Okay. The others are like the symptom assessment also, right? Finally, the phase 4 clinical trial. Like once the phase 3 clinical trial is completed, it goes to the phase 4 before, I mean, it will be approved and will be released into the market, and it will be considered as a new drug. So a drug or a molecule will be considered as a new molecule for 4 years post approval. Okay, and during this period of time, the pharma has to provide the information or the long-term safety and effectiveness of this approved drug to the regulators, that is DCGI. They will assess the risk-benefit ratio, evaluation of the efficacy, toxicity in a specific evaluation in the specific group of populations, that is the elderly patients, and they'll facilitate to integrate new treatment into the primary therapy, and it will be like cost. And normally, okay, these phase 4 trials has to be done by a pharma sponsor, right? Thank you.
So thank you, Dr. Manjunath. It was an excellent class. I think it is a bit difficult topic for clinicians like us, and you have made it quite easy. So I would say if anybody would like to ask questions directly to Dr. Manjunath is welcome.
I know it's like in these clinical trials research, it may be like in the initial stage. I mean, you may be feeling why me, or what is its the purpose? Why should I learn all those things? See, as you know, like the newer treatments are coming up, newer modalities are coming up, regimens, newer techniques of surgeries are coming up. Even the proton beam therapy is coming up. You will like anybody can ask us like what is the benefit of this newer treatments against the existing one. So where is it improving my clinical, the day-to-day life? So whether the patient's ECOG is improving, whether the physical function is improving, what is it like the newer treatments are benefiting when compared to the existing treatments? So, the research has to be a part and parcel of a clinician, right? So, we have to get into it like, हाँ हमें research करना है या फिर research के बारे में थोड़ा सोचना है, right? Thank
Yeah,
you.
I agree, Dr. Manjunath. So I would again encourage our residents and students that research is not only is a term which is only limited to a few people. It is a part and parcel of our daily clinical practice, and we have to work in collaboration with researchers, and we have to have new therapies, new treatment modalities, and they will only come out with the help of a well-designed trial, and it is a must. We should know about these trials. So I think there are not many questions, Dr. Manjunath. I think it is an offbeat topic, and I assume that the students would like to learn more about it in the future also. So with this, I thank you once again for today's class.
Okay, right. Bye.
Thank you, Dr. Manjunath.